So in today's session we are going to start our new topic, which is the isozymes. Now see, what are isozymes? They are the different variants of an enzymes that catalyze the same chemical reactions. Means a enzyme is having its different variants and they all are following the same chemical reaction there. Now isozymes are also known as isozymes and isoforms. They are the multiple form of a single enzyme obtained from different genes and thus represent the different alleles of the same genes and described as the allozymes. Now see, here we have seen a term which is the isoforms. Okay, so basically they are the isozymes are the specific category of isoforms that possess the enzymatic activity, and they are obviously variant form of an enzyme that catalyzes the same chemical reaction. Okay, so isozymes were first discovered by R.L. Hunter and Clement Market, followed by the existence of isozymes permits the fine tuning of metabolism to meet particular needs of a given tissue or developmental stage. They are the multiple forms of a single enzyme catalyzing the same chemical reaction by differing in their amino acid sequence. Okay, so difference in amino acid sequence can be known only after the sequence analysis. Okay, only after the sequence analysis, and it varies among different isozymes. Now see, basically to find difference in amino acid sequence we have to follow the sequence analysis process, and it can differ among various isozymes. Okay, then here we are having ontogenetic and phylogenetic isosomes. See, when isosomes are from the same species of an organism and they are intraspecific or ontogenetic variants. They are from different species, so they are known as interspecific variants and phylogenetic variants. Then we are having homologous and analogous variants. इसमें we are having isozymes pairing essential similar molecular structure and catalytic properties, then it is called the homologous isozymes, and here we are example having the of phospholipase. Then isozymes having different molecular structure and catalytic properties but catalyzing the same structure are known as analogous isozymes, and here is the most famous example, which is the phenylalanine and transaminase. Then the most important topic of the isozymes is the LDH. LDH is lactate dehydrogenase. It is the best isozyme to study. Basically, it is a tetramer constituted union of two types of subunits, which is H and M. Here H represents the heart and M represents the muscle. Heart is represented by a square and muscle is represented by the circle. See, basically LDH is essential metabolic enzyme, and here the term tetramer, it is the configuration is formed by two dimers of tetramer depending on how these subunits combine to each other. Okay, here are the different combinations of these two subunits generates five isozymes: LDH1, LDH2, LDH3, LDH4 and LDH5. Here we are having its subunits H and M, which is the heart and muscle. Now let's see how we have written the number of their subunits. Now see, पीछे हमने देखा था कि H को हम स्क्वायर से डिनोट करते हैं एंड मसल को हम सर्कल से डिनोट करते हैं. तो इन LDH we are having H4, तो इसमें 4 squares will be drawn. 4. Then second we are having H3 and M. H will be the 3 and wall will be the M. Similarly, we are having H2M2. 2 heart and 2 muscles. Then HM3, 1 heart, 3 muscles, and then 4 muscles, 1, 2, 3, and 4. This is the M4. So this is how we represent these isozymes. Now see, the 5 homologous isozymes of LDH catalyze the following reversible reaction, which is the pyruvate NADH H plus, and it gives the lactate and NAD. Now, the different isozymes show enzyme kinetic behavior due to their different amino acid compositions, which also provide the possibility to separate the isozymes by electrophoretic method. Now see, here we have seen the term electrophoretic method. Basically, it relies on applying an electric field to separate different molecular forms of any enzyme based on their distinct net electric charges, their size, and their shapes. Then, H4 is formed under conditions plentiful to oxygen supply and show optimal functions in aerobic metabolism. It is predominant in adult stage, it has high affinity for substrates, and it is allosterically inhibited high level of pyruvate. Followed by the M4 isozymes, they are formed under the condition of low oxygen tension and show optimal function in anaerobic metabolism. They are predominant in embryonic stages, and it has low affinity for substrates, and it is not inhibited by high level of pyruvates. Here we are having tissue distribution pattern of different isozymes. Now see, we are having 5 isozymes of all LDH, and they are differentially distributed in tissues. Now LDH1, which is having H4, it is high in heart and low in muscles. Whereas LDH2, it is high in RBC and brain but low in muscles, liver and leukocytes. LDH3, it is highest in brain and leukocyte and lowest in heart and liver. LDH4, it is moderate in leukocyte and low in heart, RBCs, brain, muscles and liver. LDH5, it is high in muscles and lower in other tissues. Then here are some applications of isozymes. Now first we are having is to access the developmental stage. Isozymes regulate the activity of a particular cell or developmental stage of embryo, like we are having LDH5, LDH4 and other or dominant in the early stages of embryogenesis, while LDH1 and LDH2 are predominant in later stage. Basically, LDH isozymes are used to scan out developmental stage. Then we are having some its diagnostic uses also, like the relative proportion of isozymes in sera, which is derived from serum. It changes during pathological conditions. Therefore, separation and identification of isozyme have diagnostic use, like serum LDH may be separated and identified by agar or polyacrylamide gel electrophoresis at pH 8.6. Then we are having some molecular markers of population genetics. This study and the cause the effect of genetics variation within and between population is called the population genetics. Isozymes are used as molecular markers for this purpose, although they have now been more informative based approaches such as DNA sequencing and single nucleotide polymorphism. Isozymes are still amongst the quickest and cheapest marker systems to identify now levels of the genetic variations, like quantifying nating systems. Now see, here we have heard two terms, which is the DNA sequencing and single nucleotide polymorphism. Okay. So बेसिकली जो हमारा सिंगल न्यूक्लियोटाइड पॉलीमॉर्फिज्म और डीएनए सीक्वेंसिंग है, डीएनए सीक्वेंसिंग इज द लेबोरेटरी प्रोसेस ऑफ डिटरमाइनिंग द एग्जैक्ट सीक्वेंस ऑफ योर फोर केमिकल बिल्डिंग ब्लॉक, and single nucleotide polymorphism is the common type of genetic variation where a single building block in the DNA is altered. So with these applications we have completed our topic isozymes.